carried out on use of microfungi in GNP production. For example, mushrooms such as Landula, Flammulina, plurals has been employed in GNP production. There are also reports on the immobilization using microbes. The microbes such as sulfate-reducing bacteria, Pseudomonas, has been used in immobilization procedure. A lot of biosensors has been developed previously using different microorganisms. Coming to the methodology, biomycelial cultures were isolated from the sterile context issue of healthy specimens of identified wild edible Tomatomyces samples during monsoon from local habitats in Goa. Tomatomyces colonies were grown on sterile Zapatrox agar. Then further morphological studies were carried out and stable and fast growing strain was produced which was further used for the inoculum preparation and pelletization. 6 days old tomato colonies were grown on sterile modified Zapatrox agar. Identical culture plugs were inoculated from each culture into 250 ml flask containing 100 ml of the modified Zapatrox solution. Flasks were kept on rotary shaker at 150 rpm and incubated at 28 degrees Celsius in dark for 6 days. Mycelial suspensions were prepared by centrifuging liquid cultures on a bent centrifuge at 5000 rpm for 20 minutes in sterile centrifuge tube. After removal of the supernatant, the pellets were rinsed with sterile distilled water and resuspended in the approximately same volume of distilled water containing 100 bits. In previous experiment, pelletized biomass from the pure cultures of Tomatomyces has been proven as a powerful gold bioreduction system. Pelletized biomass was macerated homogeneously under sterile conditions and mixed homogeneously with sterile molten 2% agar hydrogen at 40 degrees Celsius. Formed mixture was quickly poured in 96 identical wells of sterile microtest plate to obtain uniform solid bits after cooling. The beads were hardened under low temperature, and these were the beads which were obtained, which were further used for the bioreduction. For the preparation of crude fluoric acid, dried auriferous soil samples were sieved and weighed. 10 grams of the weighed sample was taken in beaker with funnel on top and transferred to the fume hood. To this, 25 ml of aqua regia was added very carefully. Then this was warmed under hot plate while stirring after every 10 minutes for 30 minutes. Then it was cooled at room temperature and decanted muddy extract with crude fluoric acid into another beaker and centrifuged and filtered and used for the screening of nanoparticles. About 80 to 90 uniform bits were immersed in beaker with 200 microliter fluoric acid. The beads were removed at an interval of 5, 10, 15, 30, 45, 16, 120 minutes and 12, 24, 48 hours and checked for the appearance of blue, purple or red color, which are the indicative of positive bioreduction reaction and good nanoparticle formation. Coming to the discussion, results and discussion. These plates, that is figure 1 and 2, are the representative plates of Tomatomyces hymen colonies which were grown on the Zapatrox agar. In figure 3 and 4, you can see the cavities formed between the mycelia. These two are the plates showing the beads which were immersed in chloruric acid, and in case of figure 6, they were immersed in crude chloruric acid. However, they were showing change in the color. They were showing the presence of purplish color. Forms of the GMP were found in case of chloruric acid, which is indicated by blue color, and also in case of crude chloruric acid. The important findings were it was possible to obtain uniform cylindrical bits. Mycelial fragments are not immobilized in same manner. A few are found at center, whereas some are found at the end. Minimum 1 hour of immersion in chloruric acid is needed to visualize the color spots or bands indicating positive GNP synthesis. The small size of bits, that is 0.42 cubic mm, permits direct visualization under microscope. Photomicrographs can be obtained non-destructively by rotating the bits on microscopic stage. Blue, purple or red color indicates positive gold bioreduction reaction and gold nanoparticle formation. Results showed that beads with a well-immobilized mycelium produce swarms of GNPs with pure and crude chloruric acid after 24 to 48 hours. The technique demonstrated that considering the 14 to 45 nanometer pore size of the agar hydrogel, the microporous nature of the immobilized viable founders, the beads are useful in production of distinct swarms of pure gold nanoparticles. Scanning electron microscopic images shows the cavities, which is like embedding a microporous fungus biomass inside nanoporous agar hydrogel matrix, where chloruric acid enters through the gel nanopores, gets inside the hyphal micromesh, and gets reduced and deposited. Future scope GNP size can be controlled by changing the agar concentration from 1 to 6%. Large GNPs can be obtained with softer agar and smaller with a harder agar. Different oviparous samples can be employed to obtain crude extracts of chloruric acid, which can be further employed in bioreduction. We are working on the enrichment of GNPs and also on the characterization. Research for further improvement to develop this technique as low cost and efficient biosensor is in progress. These are the references. Thank you.
I would request, the judges to kindly start with the questionnaire.
Yeah, very nice presentation, Miss Sujata, and also nice work. Yeah, I have only two, three, just some small doubts. This agar concentration you mentioned that you are going to try from 1 to 6%, right? Hello, Sujata, am I audible to you?
Hello, sir.
Yeah, as Sujata, very nice presentation and good work.
Thank you, sir.
Yeah, good work. Just have one or two questions, small questions.
Yes, sir.
You mentioned about that agar concentration, your last second last slide, no, that
Yeah.
you want to tune it to 1 to 6% of concentration.
Yes, sir.
Why this agar concentration is to be? I mean the ratio is to be maintained. What is the reason for that? I didn't know.
So as I have mentioned, the everything depends upon the size of the size of the agar. When the size of the agar is large, the nanoparticles of larger size will pass through it and get deposited, and we can detect the nanoparticles.
Okay, okay. So because of that, you are saying this concentration
Yeah.
1 to 6%. Okay. Then you mentioned about that beads. No, beads hardening is taken at low temperature. So low temperature is
Yes, sir.
exactly what temperature? I mean, so lower temperatures
It's uh,
like zero.
Less, yeah, sir. 0, sir. -4.
-4. Okay.
Yeah, 0 to 9.
So you are doing it with liquid nitrogen or something or something?
No, sir.
Just with sodium chloride, salt, and ice bath.
Yes, sir. Correct,
Okay.
sir.
And one more place you mentioned that this egg, no, this your soil you are no, you are mixing with aqua regia.
Yes, sir.
You are mixing with the aqua regia. Why you choose aqua regia? Why not some other acid?
Aqua regia is the only chemical which can dissolve gold, sir.
Okay, so aqua regia is the only this thing acid used for dissolving gold. Yeah. Okay, so this were my few questions. Anyway, nice presentation. Yeah, thank you.
Thank you, sir.
Sujata, nice presentation.
Thank you, ma'am.
Oh, okay. Since you are talking about bioreduction is a big word, right?
Yes,
So,
ma'am.
what exactly your nanoparticle would do for bioreduction?
Sorry, ma'am, I didn't get you.
What exactly your nanoparticles would do for bioreduction?
Actually, I'm working on fungus which can help in bioreduction.
Okay, you have just tried only fungus.
Yes,
So,
we have tried fungus and also bacteria, but in this paper I have mentioned only fungus.
okay, you have also tried bacteria.
Yes, ma'am.
So, which bacteria you have tried?
We have tried rhizobium bacteria.
Okay. Um,
I have also published the paper, ma'am, on the same.
Okay. So you have mentioned something saying that it is efficient gold biosensors.
Yes, ma'am.
So when you say efficient gold biosensors, so how much uh, effectiveness you have of your gold nanoparticles? Why you say that it is effective enough as gold biosensors?
For the perfect for the perfect efficiency, one second, ma'am. Miss, can you repeat the question?
Means, why you say that it is an efficient gold biosensor? Why you think that your nanoparticle which you have got, okay, they are efficient enough? Have you done the comparative studies?
Hello, ma'am. I can't hear you.
Yeah. Have you done any comparative studies?
Competitive study in the
Yeah, comparative studies in any other ways they have got gold nanoparticles and they have done the application as biosensors literature.
Yeah, ma'am. We have done the comparative studies already,
Yeah.
but there are no studies done on. This is the first report of use of termitomyces culture and immobilization. Means there are no other reports of fungus of this fungus in carrying out the bioreduction by immobilization.
Okay, other fungus is done.
Yes, ma'am.
This is the only fungus which is not done. You mean to say that?
Yes, ma'am.
Okay. Yeah. Thank you.
Hello, Sujata.
Hello, ma'am.
Hello, hi. So I didn't see any change in the SAM images. So how will you differentiate? Can you show me your slide of SAM images? There is no clarity in the images actually. I've seen.
Actually, this SAM image is only put in this slide to show the presence of cavity between the mycelium.
Okay, but I didn't see any change otherwise. Okay, this is it. Otherwise, nice presentation.
Thank you.
We would like to thank our judges for taking out their time for judging the oral sessions. Dear audience, now we are going to start with the next technical session.